Okay. Hello, everybody. So this is an interview with a chemist and neuroscientist named Andrew Gallimore. Some of the people listening to this may already be familiar with his work. He's received quite a bit of attention for a project called the DMTx, which is a continuous infusion of DMT intravenously designed to explore extended periods of time in DMT space. It's almost like something out of a sci-fi novel, and for that reason, it's very provocative and interesting to people. This is something that we discuss in detail toward the end of the interview. But the beginning is dedicated to his work on natural products, specifically on the stereochemistry of mycotoxin, which is one of the most potent, if not the most potent known toxins. There may be some proteins that are slightly more toxic, but if it's not the most potent, it's certainly up there. 130 nanograms per kilogram are lethal in mice. So a human lethal dose would likely be under a milligram. This stuff is extraordinary on almost every imaginable level. I was actually under the impression that the total synthesis had been completed, but I guess people have never actually assembled all the different components of the molecule. This is the Mount Everest of organic synthesis. It is the most ambitious project in the history of natural product chemistry in terms of total synthesis. And you might think, wow, this seems like a kind of bizarre and pointless academic exercise. Why would people dedicate so much energy to understanding something like this? And I think that the answer is that every natural product is extremely interesting from an evolutionary perspective. When people think about evolution, they tend to think about Darwin's finches or the peppered moth. And the reality is that while these macroscopic dimensions of evolution are fascinating and important to study, every chemical is also something that evolved. And each of these natural products is something that presumably underwent millions of years of development in the laboratory of the planet. So this is the reason that you see these extraordinary structures in nature, things that no human chemist could ever discover accidentally. And the importance in thinking about how these different natural products evolved is to some extent mirrored in the question of how our perception of reality evolved, because it should go without saying that reality as we experience it is not the true nature of reality. Color does not exist. Color is something that our perceptual apparatus has somewhat arbitrarily assigned to different wavelengths of light. Red light is not red light. It is photons that have a wavelength between 620 and 750 nanometers. But we evolved to say this is red, and we evolved to make these incredible distinctions between different wavelengths of light because it was important for our survival. There's no such thing as sound either, just oscillating gas pressure in our surroundings. Reality is a sensory phenomenon, and the reality that we experience is real insofar as it is adaptive. We have this version of reality because for millions of years, it has promoted survival and the propagation of our genes. Does that mean it is the real reality? No. One thing that Andrew Gallimore carefully articulates in this interview and in his book is that when we think of hallucinations, we shouldn't think about them as a deviation from reality so much as a deviation from an adaptive version of reality that we have evolved to perceive. And it's funny because I think for many people, especially in the psychedelic community, we are so used to hearing about the stoned ape hypothesis and Terence McKenna's hypotheses surrounding the role of psychedelics in the evolution of consciousness or language. And the idea that these hallucinations are adaptive, that we forget the fundamental, indisputable fact that in most instances, hallucinations are maladaptive. I remember going to an ayahuasca conference years ago, and there was a shaman speaking, and he said, "The reality that you see, that is not reality. The true reality is ayahuasca." And all of the white people in the audience nodded their heads in agreement. But all you need to do is spend some time with people who have schizophrenia or other disorders that promote hallucination, and you will quickly realize that in most instances, a hallucinated version of reality is not desirable. I think it's really easy when you're young and healthy and taking psychedelics to think, wow, hallucinations are awesome. Hallucinations are amazing. Hallucinations are so good. It's bad not to be hallucinating. Hallucinations are the real reality, bro. And in some instances, sure, you can have your John Carpenter, They Live experience and see the sheeple operating in their NPC world, and you know the reality. You're woke. You're Neo waking up in the Matrix. You see what's really going on. But then the LSD wears off and you go back to reality. And if you're lucky, you gained some insight, you altered your perspective temporarily or maybe permanently on this or that matter, and use it to inform your quotidian reality. But sometimes those hallucinations are damaging. I dated a woman who thought that she was Jesus and had to be hospitalized, thankfully, before we were involved with one another. I used to be friends with a guy who thought he was Jesus and had to be hospitalized. I've talked to a number of Jesuses on the phone. I've read The Three Christs of Ypsilanti. There's a lot of Jesuses out there. You don't want to be one of them. Avoid being Jesus at all costs. If you're a young person taking some LSD, it can seem great, but that is an exception, not the rule. I know an elderly woman who has Parkinson's disease. Recently, she heard her dog barking in the middle of the night. She went downstairs to let the dog into their yard and fell down the stairs and broke her hip. The dog was actually in bed with her the entire time, but the auditory hallucination of the barking dog resulted in her breaking her hip. This is why the FDA recently approved pimavanserin for treatment of Parkinson's disease-associated hallucinations, because hallucinations can be extremely dangerous. While it might seem beautiful and inspiring to take a psychedelic and have a vision of a lion or a jaguar or an alien, too much of that would result in you stressing yourself out and wasting valuable energy interacting with and maybe avoiding a threat that does not exist, or ignoring a threat that does exist. So it's important to see hallucination as a deviation from the adaptive model of reality that we have evolved to perceive. Andrew Gallimore is hoping to create a unitive model of psychedelic action, and I think he's done an interesting job. One could argue that there is something slightly tautological about the way that he is conceptualizing this issue. He is saying psychedelics are psychedelic because they disrupt our world-building model, but that is sort of similar to saying hallucinogens are hallucinogenic because they cause you to hallucinate. It's true, but it has a tautological character to it. And I don't say that as a criticism so much as an acknowledgement of the complexity of defining these things, because there are all these pharmacologically and chemically disparate substances: NMDA antagonists, kappa opioid agonists, 5-HT2A agonists, 5-HT1A agonists, GABA-A agonists, anticholinergics, CB1 agonists that exert an effect that can be characterized as psychedelic. yet these are completely different substances. We all kind of get that they're psychedelic, and one could argue that that's a perfectly compatible term for describing them. And most of the time, I don't think these distinctions are so important. Where it is important is toxicologically. If somebody is saying, well, people say that psychedelics are not addictive, but ketamine is addictive, so psychedelics are addictive, in fact. That's a stupid thing to say. If you're making toxicological claims, then pharmacological specificity becomes extremely important, because the serotonergic psychedelics are not only non-addictive, they may be anti-addictive, but the NMDA antagonists can be addictive. This is a far-ranging discussion that goes over everything from polyketide biosynthesis to DMT entity diplomacy. And I think that it covers a lot of interesting ground, especially the ground that I think many listeners are most interested in, which is basic information, how to get a good foundation in this area. This is the question that I'm asked more than anything. I want to learn about chemistry. What books do I read? I want to learn about pharmacology. What books do I read? And of course, I could recommend Goodman and Gilman's The Pharmacological Basis of Therapeutics, or Daniel Perrine's The Chemistry of Mind-Altering Drugs. But I think that Andrew Gallimore's new book, Reality Switch Technologies, is a great intro. So I recommend it, and I hope you enjoy this conversation. It's well known that the cannabis plant contains a wide variety of fascinating phytochemicals. It's one of the most studied plants in the world. And so I'm always amazed when researchers discover new compounds in cannabis. What was especially amazing is that in 2019, a group of researchers in Italy found a new cannabinoid called THCP that is structurally very similar to THC, but it has a 7-carbon chain instead of a 5-carbon chain. The extension of that chain gives THCP much greater potency. So at the CB1 receptor, which is responsible for the stoning effects of THC, THCP binds with 33 times higher affinity, making it the strongest known phytocannabinoid. I thought that this would remain an obscure scientific curiosity, and I was amazed to find that within 3 years of its discovery, techniques had been developed to industrially produce THCP from hemp. I saw that Canaclear.com was selling it. I independently ordered a sample, analyzed it via NMR, and found that the material was bona fide. So if you are interested in THCP, delta-8 THC, HHC, or any other unusual phytocannabinoids, go to Canaclear.com. All of their materials are third-party tested for quality and compliance, and if you use the code Hamilton, you will get 15% off any purchases. Thank you, Canaclear. This podcast is also brought to you by Lucy Nicotine, a company that makes nicotine pouches, nicotine gum, and nicotine lozenges. I particularly enjoy the apple ice flavored nicotine pouch. It is a refreshing and well-formulated product. If you don't already use nicotine products, I recommend you don't begin. They're habit forming. But if you do, I think this is the finest nicotine product on the market. Thank you, Lucy. This podcast is also brought to you by matcha.com. Grab a traditional matcha bowl or your favorite mug and enjoy a hot cup of matcha first thing in the morning. I don't drink coffee, but I think matcha is a wonderful way to start the day. It contains L-theanine, it contains caffeine, which is what I'm most interested in, and it has all sorts of purported health benefits. Matcha.com was founded by psychedelic pioneer Dr. Andrew Weil. All of their matcha is imported from Japan and third-party tested for heavy metals. It's delicious, probably healthy, and certainly stimulating. So enjoy a cup of matcha from matcha.com. I love your book.
Thank you very much.
I think it's awesome. I think it's a sorely needed text. It sort of addresses one of the questions that people have the most, which is always, where do I get started in this? How do I begin? What's a good introductory text? And I was always recommending these books from the 70s or 80s or 90s.
Yeah.
And so it's really great to see an updated text that is oriented toward psychedelics, which is what most people are interested in, at least that I interact with. So to have something that's totally focused on the pharmacology and neuroscience and chemistry surrounding psychedelics, I think it's great.
Yeah. Thank you. Yeah, that's what I tried to do. Because I knew there was a missing book there. I mean, I was reading books like Solomon Schneider and things from like the 70s and these Lumpanics books and stuff that just weren't cutting it, in my opinion. So I thought, yeah, I need to write this book. No one's written it before, unless you want to dive into the really academic stuff where they spend chapters and chapters on binding constants and stuff, which most people don't want to do that. They want to really kind of develop a kind of a deep understanding of what's actually going on with these molecules when they get in the brain. So that's why I wrote the book.
Yeah, I think it's great. And I think just to be clear, it's available now, right? People listening to this can order a copy of it on your website if they're interested.
Yeah, or Amazon, anywhere basically. Yeah,
okay.
Widely available.
Okay, all right. Well, that's good. Yeah. So I mean, I'm curious about the motivations for this book, but I also wanted to maybe talk about your work with DMT because that is, I think, what you're best known for. And I don't really know all that much about how you got to be working in Japan or any of really the backstory of your work in this area. So if you could just begin by telling me a little bit about who you are and how you got started in this area, I think it would be interesting to know.
Yeah. Have we started, by the way? Is this the preamble?
Oh no, this has begun. Yes.
All right. Yeah. Well, I started. I actually began as a chemist. My kind of originally when I first went to university, I studied chemistry. Before I went to college, when I was kind of 16, is when I first kind of discovered psychedelics, and was first introduced to Terence McKenna by a friend at school, an interview in the back of a magazine, and he spoke about DMT, and I read about DMT, and I thought, yeah, this is really cool stuff. So I was 16, 17 at the time, so I knew I wanted to go to university and learn about psychedelics and about drugs generally. So I was originally going to study pharmacology, but then a professor said, no, you should study chemistry. It's going to stand you in much better stead in the future. And I think that's good advice as well. If you're interested in drugs, study chemistry, for goodness sake. That's really important. And so, yeah, I studied chemistry. And even at that early age, I was kind of known amongst my friends as this guy who was really interested in drugs. And then kind of in the final year of university, my master's year, I took this course, natural products chemistry. And kind of weirdly looking back, I had no idea what this course was going to be about. I didn't think it was going to be of much interest. But then we started learning about like the biosynthesis of terpenes and alkaloids and polyketides, and thought, wow, this is, I kind of fell in love just looking at these beautiful biosynthetic mechanisms, these couplings and rearrangements and cyclizations and all this kind of stuff, building these really intricate and beautiful molecules. And I thought, wow, this is it for me. It looked like kind of chemical poetry to me on the paper. I thought, yeah, my PhD is going to be in biosynthesis, in polyketides particularly.
Oh, wow.
Yeah, I don't know if you know this molecule, mitotoxin.
Of course.
Yeah, of course. It's the most like, it's like I remember seeing this beautiful molecule. People who don't know it, it's the largest and the most toxic non-proteinaceous molecule. It has this polycyclic ether ring structure. I forget how many rings there are, but it's such a beautiful thing. And I thought, I want to work on mitotoxin, just completely randomly. And so I found Jim Staunton's group at Cambridge, And I said, okay, I'm going to work for Jim Staunton. I'm going to research polyketide biosynthesis for my PhD. Unfortunately, Jim Staunton was just on the verge of retiring, but his group had been taken over anyway by Jonathan Spencer. So I went to work for Jonathan Spencer in the Department of Chemistry at Cambridge and studied polyketides, biosynthesis for my PhD. I actually worked on mycotoxin towards the end of my PhD. I developed a retro biosynthetic analysis. So basically try to work out how is this huge molecule constructed inside these marine organisms, Gambia discus toxicus, this dinoflagellate, and developed this model. And we actually found a discrepancy in the structure. We worked out how it should be constructed, and we found that one of the ring junctions had the opposite stereochemistry than it should have. So we developed, basically, it's a little bit of a story, but we developed this, what we call the stereochemical uniformity rule, which basically says that any single polycyclic ether must be constructed from a stereochemically uniform polyepoxide for enzymatic reasons. And this rule, we did analysis of all the different polyethers, and we found that this rule applied perfectly to all of the marine polycyclic ethers. But there was one problem with one molecule, and that was mitotoxic. It had this ring junction that was incorrect. So I showed my supervisor this. I said, this doesn't look correct. And he said, well, go back to the original assignment. Look at the original assignment published in JAX, I think it was. So I went there and I looked, and they had this one little paragraph that said we had particular trouble assigning this part of the molecule in exactly this ring junction that I'd identified, called the JK ring junction. So we wrote this paper and published it in Angewandte, Stereochemical Uniformity in Marine Polyether Ladders, Implications for the Biosynthesis and Structure of Mycotoxin. And the paper was kind of an essay, like a theoretical model, as we didn't have any experimental data. They sent the paper to Duilio Arrigoni for review, and to some Japanese guy, we think, having read the referee report. Duilio Arrigoni absolutely fucking loved it. He said it should be published without a single alteration, but this Japanese guy hated it. Said, how dare you? Because it was a Japanese group that assigned the structure of this molecule. So, how dare you kind of question the work that has taken years for this group to assign the structure of this molecule, the largest non-proteinaceous molecule. We just kind of said that maybe it was wrong. So they didn't know what to do. They had these completely opposite kind of referee reports. So then they sent it to KC Nicolau, who we know as Nicolau because he suggested we reference KC Nicolau. And then he really liked it. And so eventually we got it published anyway. So that was kind of, the kind of the combination of that original kind of falling in love with polyketides at the end of my degree to actually kind of working on this beautiful molecule. And it's still, the jury is still out, by the way, on this JK ring junction in mycotoxin. See if you can spot it. If you look at the structure, you'll see that one of the ring junctions looks different to the others.
Oh, wow. That's fascinating. I had no idea that you worked on that. That's really cool.
Yeah.
That's amazing. And so in terms of the synthetic mitotoxin, does this mean that the total syntheses that have been done are potentially incorrect? Or what was the reference material that you were using in your analysis? Was it isolated from a natural source, or was it some of the synthetic material?
So the total synthesis isn't there. I mean, Nicolau has constructed bits of it. So there's four separate ladders, if you like, four separate these contiguous ring structures in mycotoxin, and Nicolau has synthesized pieces, of this over the decades, really, but no one's put the whole thing together yet. So there's no pure synthetic mycotoxin. So what we did was a purely theoretical analysis. We looked at all the different examples of these marine polycyclic ethers. We looked at brevetoxin, ciguatoxin, yesotoxin, all of these things. They all have the same kind of structure in common. And we did this retro-biosynthetic analysis. We took them backwards to their hypothetical polyepoxide precursors, and we found this stereochemical uniformity rule. Every single one was constructed from a stereochemically uniform polyepoxide. So it was all RRRR or SSSS epoxides, basically, all from oxidized from the same face. And we thought that made sense because you probably got a single monooxygenase, a single epoxidase enzyme that's doing, kind of iteratively through along this long chain. So we thought it doesn't make sense that there are any exceptions here. And we were correct. And we also found an error in a small polyether, a polycyclic ether called Brevinal, which is smaller. But we also found that one of the rings was also wrong there. And then about a year after we published this analysis, they actually reassigned the structure and said we'd got that wrong. So it's proved, this rule that we developed has kind of proved a little bit useful in assigning these. They have to be derived from stereochemically uniform polyepoxides. But with mitotoxin, it was purely a theoretical analysis. But then Casey Nicolau actually did some synthetic work on the back of our paper. But he only synthesized the correct version of this part of the ring. He didn't synthesize kind of the incorrect version. So he didn't kind of compare the NMR of both different types to see, which was most likely to correspond to the natural product. So we're not 100% convinced that this analysis that Casey Nicolau did is kind of conclusive. So yeah, it's still in the air. It would be really cool after, that was 2006 that paper was published. It would be really cool at some point if we could be proved correct and kind of vindicated. But yeah, we just have to wait and see. But yeah, that's my work with polyketides. But I left that behind.
This podcast is also brought to you by the Apollo. The Apollo is a wearable bracelet or anklet, whichever you prefer, that produces a vibrating frequency that can achieve a number of different effects depending on how you set the device. It can be stimulating to wake up in the morning or relaxing at the end of the day. Sometimes I wear it while I'm sleeping. Sometimes I wear it while I'm driving. I find the effect really nice. It's sort of like strapping a purring kitten to your ankle. And I know that it sounds a little bit weird, but if you enjoy massage, if you enjoy relaxing, vibrating chairs, I think that this is in that vein of an effect. It feels really nice and calming, and it's a great way of non-pharmacologically altering your mood. This is a product that was created by a psychiatrist named Dr. David Rabin after years of research, and I think it's a really interesting device. So if you want to learn more about the Apollo, go to apolloneuro.com. Thank you for the sponsorship, Apollo. Was it, because I remember meeting a chemist who had done his PhD at MIT in polyketide total synthesis, and it had sort of driven him insane. After he got his PhD, he never did chemistry again and became a lawyer. And and I can imagine that there's something almost maddening. I mean, this is really like about as complicated as it gets in terms of natural product chemistry. I mean, I don't think there's anything else that rivals these polyketides. Did it burn you out? Did you?
No, I mean, I didn't do, and I really wasn't, I'm not really much of a synthetic chemist. I mainly worked on basically doing genetic manipulation, so knocking out genes from the biosynthetic gene cluster of these polyketide producing organisms, and then then isolating the metabolic products, the kind of the mutant natural products, then trying to work out what these genes do. So it's a lot of really tedious, get liters of this black sludge that you would isolate from these streptomyces cultures, and then extracting it down and doing, prep HPLC and stuff, and getting down to this pure white powder from, gallons of this blacks, it's really tedious crap, not fun at all. Then you obviously you'd end up with 1/4 milligram of this white powder, which would then go to the NMR section, and you would start running your NMRs, and then the fun of trying to assign the NMR. You got these large structures, all the the carbons. They're kind of repeating structures, right? So there's a lot of overlap in all of the proton and carbon signals. So you're looking through these nosy and all these different and trying to find where are the differences between the natural product and this molecule that I've isolated, and it might be just a shift in stereochemistry on one ring or something. It was clear to me that I wasn't going to be doing this for the rest of my life, and I wanted to move theoretical. That's when I made the shift to neuroscience.
Oh, just one more question.
Yeah.
Polyketides.
Go on.
I didn't know that you did any of this. And these are also some of the most potent known poisons. So did you take any special precautions when working with these chemicals? What is the protocol when you're doing the isolation of these compounds and working with them?
So mycotoxin in particular, as I said, it's astonishingly toxic. You have to take special precautions. I mean, you're wearing a mask, you're wearing gloves. We were never isolating very large amounts. So it was always,
you don't need to.
You don't need to. You're right. Yeah. But it's weird. There was kind of a disconnect there. I mean, it's not like working with viruses or bacteria where there's a danger of it kind of escaping as such. But there is a personal risk, certainly. But you get used to, as working in a chemistry laboratory, you get used to taking precautions and making sure you're not splashing things on yourself.
Although in the realm of small molecules, it's probably closest to a virus of almost anything. I think that ciguatoxin is the only substance that I'm aware of that is a small molecule that has been associated with sexual transmission. Have you seen that paper?
No. No, I haven't.
Sexually transmitted poisoning from ciguatoxin.
So it gets into your semen? Is that
is the claim in this paper?
Wow. Okay. Yeah, I should have thought about that. That might explain a few things. Joking.
That's really cool, though. Okay, so you were doing this immensely difficult work, but you knew that you wouldn't continue with that for the rest of your life, and you were still interested in psychoactive drugs throughout all of this work, I assume.
Oh yeah. So I was still that was always the aim, weirdly, even though I'd taken this weird little detour into natural product chemistry. It certainly wasn't unrelated, right? I mean, the molecules that I'm particularly interested in, these psychoactive molecules, are natural products. But I'd taken a detour into really toxic products instead, really complicated as well products. But that stood me in good stead, getting a feel and an understanding, and really just developing a sense of the beauty of these things. I mean, these molecules are just, I don't know. If you're not a chemist, it's maybe difficult to understand, but you will understand. When you look at a complicated, complex molecule, it just sends a little shiver down your spine sometimes, and you think, "Wow, that is just fucking amazing." This poetry that these organisms are composing with these molecules, and we still don't understand why. I mean, why does Gambia discus toxicus make this incredibly large, ridiculous? I mean, what's the mass of it? It's like 1,500 or something. The molecular mass of this, it's massive. The amount of energy and time and resources required to construct this molecule, and there are more efficient ways of making toxic molecules with these huge ring systems. So yeah, that's I never lost the love for for drugs at all. In fact, it probably enhanced it. But I knew I needed to kind of divert myself back to a more direct working with psychoactive. So that's when I left my kind of polyketide work and diverted to computational neuroscience, which is a bit of a shift. But yeah, that was the time.
And so you were in the UK doing your PhD.
Yeah, so I was at Cambridge, did my PhD, then I stayed in Cambridge and continued to work on polyketides. So again, more in the biochemistry, or shifting more towards doing more genetic manipulation, trying to create novel polyketides by manipulating the biosynthetic gene cluster, with the ultimate aim being that you would generate in vivo these novel polyketides that might have pharmaceutical or other interest. But I became pretty tired with the speed of it. It's a very slow, tedious process. You have to do these genetic manipulations, which take a long time, and they don't always work. Then you have to isolate. So I don't know.
Yeah, and the structure activity relationship is impossibly complicated. I mean, it's hard enough looking at something like tryptamines or ergolines to have a comprehensive understanding of the structure activity relationship. How are you going to understand the significance of a single methyl group or oxygen in a polyketide where it's just bottomlessly complicated?
Yeah, exactly. Yeah. So I decided that was enough. I'd made my contribution, and left at least one kind of fairly large open-ended question that Casey Nicolau can deal with. I mean, that's his thing. I'm not going to be doing total synthesis of anything. I wasn't very good at synthetic chemist. My yields were always appalling. So I realized that wasn't for me. Yeah.
So then computational neuroscience.
Yeah. So then I kind of felt that I studied, as well as chemistry, I also studied pharmacology. So I had that kind of low level covered. I knew the chemistry, I knew the base, the low level pharmacology, the drug receptor interactions, subcellular kind of stuff. But I knew there was some kind of gap at the high level, the actual neuroscience level. So I needed to fill that, and I thought about ways to do that. How could I shift into a postdoc that was not so wet, so not practical. I didn't want to be working with rats and isolating neurons from rats or doing patch clamping. I worked in one of these labs for over a summer and absolutely fucking hated it. I hated killing rats.
Yeah.
And I just doing this wet stuff again. I found myself, oh my God, I'm going to be dragged back into this wet science, which I didn't like. So I thought, okay, I need to steer myself away from that. And computational, this to me is where in many ways, is where it's at now. We kind of understand enough about the way that neurons behave and the way that neurons interact, that we can apply this new computational technology that we have. So that it was clear that we were in the beginning now, or more than the beginning, but actually really deep into territory where computers could be used in a really good way to understand what's happening inside the brain, at both the subcellular level, so systems biology, where we're looking at the emergent properties of these complex subcellular networks of signaling molecules and how they influence the behavior of neurons. So I worked on that, and also network level. So how do large networks of model neurons generate and share information, and how does that relate to the function, the various functions of the brain? So that's what I did. So I did a postdoc at University of York and then University of Oxford, And then I got a position in Okinawa in Japan. My contract ended at the University of Oxford, and I thought, what the fuck am I going to do now? And then there was this job opening, this professor in Okinawa who was just happened to be working on the same kind of thing I'd been working on at Oxford. And
And
so
what is that specifically?
I was looking at subcellular models of synaptic plasticity. So particularly, how do the synaptic connections change? What is the molecular underpinning of the changes in the synaptic connection? How does it change over a long period of time in response to neural activity, basically? So it was more like systems biology, computational systems biology. So I was looking at modeling these networks of signaling molecules and looking at their emergent properties, trying to understand how the changes in synaptic strength emerge from these signaling molecules. And of course, that relates also to psychedelics, because that's what you've got going on. You have a molecule that's binding to a receptor on the surface of a neuron, and it's manipulating, it's perturbing this complex intercellular signaling network, which ultimately has the effect of altering the function and behavior of the neuron.
We got the rectal kratom guy right off camera right now. I mean, this is what you're telling me. I don't know. That's boofing kratom. It's become a meme on the internet. This is a kratom tea product. It's not intended for boofing. Even if the pH of the human rectum is between 7 and 8, and the pKa of mitragynine is 8.1, that's just not really relevant here. This is a tea consumption in hot water. That's what it's for. That's what it should be used for. If you want this tea, you can get it at toptreeherbs.com.
Okay. So yeah. then what is the lab in Japan doing? Because I remember you saying something about them doing work with MDMA and octopuses. Am I imagining this?
I wasn't. No, you're not imagining it, by the way. But it was LSD in octopuses.
Oh, LSD in octopuses.
Yeah, it wasn't me doing that work, but there was a Japanese guy in my university in Okinawa who was giving LSD to octopuses. And I don't know what he expected. I think he expected them to do some kind of weird dance or something, but they didn't. I think they just kind of did this, like hunched up, like scared, I guess. I mean, octopuses are smart, aren't they? So you can imagine them getting kind of a bit terrified about, what's happening here when you give them LSD. So perhaps it's not so surprising. I forget the name of the guy now, but you can probably find his papers. But unfortunately, it wasn't as interesting or exciting as he hoped when he gave them LSD. But yeah, he was doing that in Okinawa. Yeah, I don't know if he still is, but he was at one point a few years ago.
Was the lab that you were joining also involved with psychedelics, or this was just another place to do computational neuroscience work?
Yeah, it was just another place to do computational neuroscience work, really. There aren't many labs, as you can imagine, in Japan that are doing, certainly doing, work in humans. That basically doesn't exist in Japan yet. Hopefully it will do at some point in the future. The Japanese are more open to psychedelics than most people give them credit for. But yeah, I just wanted a really good kind of prestigious lab that was doing solid work in computational neuroscience, and that was what came up. I'd wanted to live in Japan since I was a teenager, kind of the back of my mind. I don't know why, but I've always been fascinated by Japan and Japanese culture. So this was it was a fortuitous situation, really, that my contract had ended in the UK and that I needed a job. And this came up. I've been here ever since. I now moved to Tokyo, but I'm no longer in the academy, so to speak. I've now gone fully independent, left academic science.
Did you have friends in Japan? Did you speak Japanese, or did you come to... No?
No. I didn't speak any Japanese. Well, I tried to learn when I was a teenager, from books and tapes. This was the tapes era, cassette tapes, but no, I'd forgotten most of that. And no, I didn't know anybody. Fortunately, the university has a really good kind of support system. So they don't just drop you in at the deep end. They support you in getting bank accounts and, how to go to the supermarket. But it's a huge culture shock. I mean, you have to contend with the fact that you're going to be illiterate when you arrive, basically. There's no way around that. You're going to be illiterate. You can't read anything. You can't read what's on the packets in the supermarkets. You can't read the road signs, although they're usually in English as well in Okinawa. But you can't go to the government office and try to deal with some minor issue. You can't call your mobile phone company, and it's like you need support for everything. And Japanese is a really difficult language to learn, which I'm still dealing with. After 7.5, almost 8 years here, I'm still not kind of native fluent at all.
Yeah, I can't imagine. I have so much respect for people that study science in a country other than their own in a foreign language, because it's hard enough running instruments, using these often extremely non-user-friendly programs in your native language, and then to apply all of that to a foreign environment, foreign language. I can't even begin to imagine how difficult it would be.
Yeah, it's pretty tough. Fortunately, the university was actually an English-speaking university, so it's a very international university.
I see. Okay.
It's not like one of these universities in Tokyo or in Kyoto, where you really do have to, everyone speaks Japanese, but it's still difficult day-to-day life, living in a foreign country where the culture and the values are all completely different in the UK. But I love it here now, so I kind of don't plan on going anywhere soon.
So I think it's really cool. So you're doing the computational neuroscience work in Japan, and I was looking over your publications. I don't know exactly at what point you started publishing on psychedelics, at least in the around 2016, 2015, maybe even earlier. Is that right?
Yeah, a bit earlier. So I think I published a paper in a bit of a fringe journal. It was really my first paper that I published on psychedelics, which is called Building Alien Worlds, which is the neuropsychological and evolutionary implications of the astonishing psychoactive effects of DMT, where I looked at. So basically, I was interested in, why is it that you can take this small molecule, this very simple, relatively simple certainly compared to what I'd been working on in Cambridge.
Yes.
This really, you look at DMT, the structure, as you will understand, and it looks like a blunt instrument. You think, well, this isn't going to be interesting at all. The amine is masked by these two methyl groups. There's no functionality on the indole, and you think this might have some mild effect, but you couldn't be more wrong. When you look at this molecule. And so that, to me, as a chemist and a pharmacologist, always struck me as uncanny, that this really very, very simple molecule could reliably elicit such astonishingly profound effects on the brain. You can actually change the structure of your reality in ways that you could never have anticipated, or you can barely even comprehend on the way out of the experience, what just happened. So I thought I need to really turn my pharmacology and my neuroscience to DMT, really think about what's actually going on here when you take DMT. So I spent a lot of time thinking about it, thinking about the way the brain constructs your model of reality, your world model, as I call it, and how perturbing these particular sets of neurons in the brain with DMT, how it can actually cause the world that emerges from neural activity, the world, your subjective world, which is this emergent model, how that can actually change. So I wrote this whole paper, the implications of what was happening with DMT. And I published that, and that was refereed by a scientist called a psychologist called David Luke, who you might well know. I mean, he's very big. I mean, he organized Breaking Convention. Of course, you were there. That's when I first met you, and he refereed it, and so I was on his radar, and he's been wonderful actually to me. I was invited to speak, or I was accepted to speak at that Breaking Convention in 2013. And really it kind of went from there. So that's when I first became, to some extent, known within this little clique that we live within. And then I continued to write mainly about DMT, really, but also all other psychedelics as well, trying to avoid getting into the woo and the new age stuff, whilst also avoiding being kind of trapped in kind of restrictive, orthodox, scientific kind of thinking, but trying to kind of have one foot firmly within the scientific arena, maintaining a neuroscientific underpinning to everything that I'm talking about, whilst reaching out with the other hand into territories that other people don't often like to go, or afraid to go, or are worried about their funding and stuff. I have this freedom in that I'm not concerned about my grant applications. I'm not concerned about tenure. So I'm able to talk about things and say things and speculate on things that other people wouldn't do. And so that's what I've continued to do, really, is to think about, what really is going on with DMT. What the hell is it? What are we to do with an elf, as Terence McKenna used to say? What is going on? How is it possible that you can get this 100% reality channel switch by perturbing your brain with this simple molecule? And why is that different to 5-MeO DMT? Why, when you stick this methoxy group in the 5 position on the indole, do you get this completely different behavior? Why is psilocybin different? Why? And then you get things like salvinorins and the NMDA antagonists and the tropanes, which are also wild in their own way. All of this is really fascinating and exciting to me. So really, this kind of ultimately culminated in my new book, Reality Switch Technologies, where I try to provide this unified framework for understanding psychedelics that anybody without even anyone with a high school biology education, I think, should be able to pick this book up and develop what I consider to be quite a deep and fairly thorough understanding of what's actually going on when psychedelic molecules enter your brain.
Yeah, and one thing I really like about the book is I think it, from the outset, addresses one of my biggest questions about psychedelics, which is people get so caught up in essentially moral dimensions of psychedelic experience. Is it good? Is it bad? Is it healthy? Is it dangerous? Is it spiritual? Is it not spiritual? That they often don't take a step back and say, well, we're just taking for granted the fact that this is even possible. There's no, as far as I'm concerned, there is no biological reason that we would necessarily assume that the psychedelic experience would have to exist. That this would be a necessary ramification of the brain or consciousness existing. But I think it becomes more clear when you clarify the fact, which you do in your book, that reality itself is a sort of hallucination. Reality doesn't exist to begin with, as we conceptualize it with our senses. And so when you recognize that reality and our experience of external reality is itself a construct and a hallucination, any sort of deviation from our typical perception of that external world begins to feel, it makes more sense, because you're really thinking about it in terms of adaptive or maladaptive representations of reality, as opposed to accurate depictions of reality, because there's no such thing.
Right. Yeah. So that's exactly what I always try to make clear. I try to work with what we know. I don't talk about, or I try not to make that a differentiation between real or not real, right? Is it real? That old question we're always asked: is the DMT experience real? Nor do I talk about consciousness as such. We know that we are conscious. We know somehow that this structured information that emerges from neural activity has this subjective component. We don't really understand why that's the case, but it's clearly the case. And we know that if you manipulate the information being generated by your cerebral cortex, that you get a change in the structure of your subjective world. So we know there's a relationship there. We don't know what exactly that relationship is or why there's even a relationship, why it has subjectivity, but we know that it does. So I focus on, okay, the world that we experience is always constructed as a pattern, a highly complex emergent pattern of unified information by the cortex. That's what we know. Sometimes that has a relationship to the environment. There's clearly an adaptive mapping between what's going on in the world you experience and what's going on in the outside world, because that's, we evolved, our brain evolved to construct this model of the environment that works. We don't know if it's true. We know that it's adaptive. We know that it helps us to survive and to reproduce. So I always focus on the model. The world you experience is always a model, always being constructed by a brain. And that includes the DMT world. You can't experience a world if the brain can't construct it. So questions about, is the DMT world, is it real or is it all in your head? That old, completely false dichotomy, right? Even if the DMT world is mapped to some external, extrinsic environment, some other reality, some other dimension, whatever, some other universe, your brain has to construct a model of it for you to perceive. If your brain can't construct a model of it, you can't experience it. So what is happening with DMT is it's causing your brain to construct a different model, a model that, for reasons we don't really understand, bears no relationship whatsoever to the normal waking world, which is a model of the environment. So it appears to be this entirely non-adaptive, completely crystalline, clear, almost, often quite stable and reproducible model the brain starts constructing when you perturb it with DMT. That's what's happening. It's a separate question then. Is that model just like the normal waking world model? Is it mapped to, is it receiving information from, is it being modulated by, is it being tested against some external environment? That's the question. That's the question. That's the reality. That's the ontology question, in my opinion, because the world is always in your head. When you're dreaming, you're also building a model in your head that normally looks very much like normal waking world. And the DMT world, just like that, it's being constructed by the brain. But that raises some really interesting questions, which I discussed in this 2013 paper, which was why, the brain evolved to construct your world as a model of the environment. It evolved, it takes millions of years of evolution up to the level that we're at now, where the brain is able to construct this perfectly stable and adaptive and informationally rich model of the environment. That took a lot of time, and yet suddenly when you perturb it with this molecule, it's like it suddenly switches to building this model that you think, when did the brain learn to do this? Why is it possible that your brain can suddenly start constructing these extremely complex hyperdimensional worlds filled with extremely powerful and intelligent beings, and everyone starts reporting seeing the same types of beings, seeing the same types of worlds, passing through the same kind of structures, the same kind of dimensionality? That to me remains to some extent confounding. I often describe it like a British child, a young British child who only speaks English, suddenly one day flipping and speaking fluent Siberian Yupik or some African click language. You go, where the fuck did he learn that? That doesn't make any sense. And it's a similar kind of problem, I think, that we have. Why can the brain how does the brain even know how to construct these models? And why would perturbing it with DMT cause it to do so?
Do you have an answer?
You're waiting for the answer. Well, I don't. I don't have a definitive answer. So one thing I suggested in the 2013 paper was that in fact there was a period of neural evolution, that in fact DMT could have been some kind of ancestral neuromodulator. And this was back in the day, well, only 9 years ago, but back in the time when people were taking seriously the idea that the pineal gland might actually be generating DMT. Now we know, actually, Dave Nichols did some excellent analysis of this, and I think the consensus is that the pineal gland is much too much of a small, tiny little structure the size of the end of your little finger, and then it's probably not pumping out DMT in large quantities. But I thought about the idea that DMT might have been produced according to a diurnal cycle similar to melatonin, and that actually when you sleep at night, in ancient times, your brain was actually producing DMT, and so during that time your brain would have essentially be learning to construct this alternate reality, whereas during waking hours you'd be within the normal waking world and your brain would be constructing that model of reality. And so it would be like a parallel kind of a parallel neural evolution, if you like, and that would explain it. I'm no longer that convinced of that. I mean, I was never that convinced in the first place. It was just a hypothesis. It was an idea. But
right, but this speaks to some need for biological justification, which has existed throughout the history of psychedelic research. These things must be produced to account for dreaming. These things must be produced to account for religious or near-death experience or the hallucinations of schizophrenia. They must have some purpose. Maybe they exert a neuroprotective effect, and that's what the near-death experience is. And I'm not saying that that's necessarily false, although I agree with you that the evidence for it is becoming increasingly weak. People have looked at this pretty extensively, and in the realm of schizophrenia, it has not translated into any research that is taken seriously or has stood the test of time. In the realm of dreaming, I think the same is true. In the realm of near-death experience, it hasn't been adequately investigated, but the evidence is not hugely compelling, although there is a little bit there. But this still doesn't account for all of the other substances that fall under the broad umbrella of psychedelic, with anticholinergics or kappa opioid receptor agonists or NMDA receptor antagonists. And so clearly not all of these things are endogenously produced signaling molecules. So it doesn't seem to account for it. It doesn't seem to be the best explanation that these are all systems that exist to be stimulated by endogenous psychotogens or whatever you want to call them. So the question remains, why are we able to have these experiences?
That question still remains, yes. And you're right. So people have said, DMT, maybe it is responsible for schizophrenia, the transmethylation hypothesis, right? Going back to the 50s, I think, the idea that your brain is methylating these tryptamine metabolites and producing DMT endogenously, and that is disrupted in schizophrenics. So they're producing too much DMT, and people have looked at that. There's no reliable difference. Some papers show some significant difference, but there's been no, as you say, there's no real clear evidence that schizophrenics can producing more DMT than normal people. And then we get the dreaming thing. Jace Calloway back in, well, I think it was the 80s now, who said maybe that dreaming is caused by DMT. Nice idea, nice hypothesis, but again, it's not substantiated. People have looked, by the way, at levels of DMT over a 24-hour period. You don't see that spike during nighttime or during REM sleep that you would expect. So no, I don't think it's dreaming. What else? Yes, hypoxia. That's kind of separate, because that deals with protection of cells in hypoxic states. protects against cell death in neurons when you're having a stroke, for example, or being asphyxiated. But that itself seems separate to the psychedelic experience, although it would explain NDEs. If you are on the verge of death and your brain is being starved of oxygen, producing DMT would be the time to produce it. But why then that produces this psychedelic state, that is a completely different question. So yeah, we're continually trying to find a simple, easy to accept explanation for what's going on here. But there isn't one, I don't think. You're right. With the kappa opioid agonist, who would have thought that this single molecule, or these terpenes that are produced by this single plant, this extremely rare plant, really, in the wild, that only grows in the cloud forests of southern Mexico or something, that just happens to produce as well this reality-shattering molecule of salvinorin, and that it does so, that it's not an alkaloid, and that it does so via the kappa opioid receptor. I mean, this was like ridiculous. As you understand, many people don't understand how fucking weird salvinorin is as a molecule. It's constructed in a completely different way to the alkaloids, not derived from tryptophan or from any amino acid. It's derived from this terpene pathway, which is completely separate. And it doesn't work via the serotonin system at all, it seems. It works by binding to these kappa opioid receptors in the claustrum, this small part of the brain beneath the cortex. So it makes you wonder what other mechanisms. I mean, the book, I talk about 4 basic mechanisms, what I call the classic mechanism, which is, of course, your 5-HT2A agonism. Then you get your tropines, which are muscarine receptor antagonists. Then you have your NMDA receptor antagonists. And then finally, you have your kappa opioid agonists. And it makes you wonder, are there other mechanisms that we simply have no idea about, that we couldn't predict, that are sitting there in the brain, this bizarre, maybe this kind of unusual, unstudied orphan receptor that when you stimulate it, switches your reality channel to somewhere completely different that no one's ever anticipated before. I mean, that's kind of exciting, right?
Oh, of course. I mean, and also there's GABA A agonists as well, which are
right.
Absolutely. I mean, again, the word psychedelic lacks a certain specificity, but
yeah,
I've used high doses of gaboxidol, and that is as psychedelic as anything else. It's different, of course. It's a different type of experience entirely, but that same sort of proliferation of ideas and perceptual disturbances is very much present. It is not in any way analogous to a benzodiazepine. This is something that is visionary and completely alien and strange.
Wow. No, I've never tried anything like that, so I've not experienced it.
It's worth trying, not because it's enjoyable or good. It's also not bad either. Actually, funny story, the most intense gaboxidol experience of my life was in Japan, because I was filming at a robotics conference in Japan.
Obviously.
on very, very short notice, and I had to start filming the day that I landed. And so I thought, all right, I've got to hit the ground running. I'm going to take an Adderall first thing in the morning to reinforce this new sleep schedule, and at night I'm going to take gaboxidol at a high dose to knock myself out. And I'd taken gaboxidol at lower doses many times before, and I'd had mild effects from it. But because I was taking it at the equivalent of something like 2 p.m. New York time, I was much more awake and alert, and unintentionally had one of the most intense psychedelic experiences of my life, taking this stuff that I was thinking was going to just knock me out. And then I had to work the next day. And not only had I barely slept, I'd spent the entire night in this visionary state, trying to trying to figure out a way to lose consciousness, but instead I was hyper conscious, and it was something I couldn't explain to anyone else that I was working with. Like I took this conformationally constrained muscimol derivative, and something really, really weird happened that I didn't anticipate. But yeah, it seems that there's almost no receptor system in the brain that I'm sure there are, but it seems most of the major ones can be modulated in some way to produce an experience that could be classified as psychedelic.
Yeah, it's fascinating, isn't it? You have these molecules that at certain dose levels they manipulate neurotransmitters in the way that we understand and expect. GABA modulators, we understand benzodiazepines, right? But then when you shift and you start pushing the dosages, you start to see unanticipated emergent effects going on, whether it's caused by activity at the same receptor, and that you're getting some shift in the emergent effects at the network level, or whether you're starting to tickle other receptor subtypes, and getting a completely different effect that way as well. Often that's not clear and requires quite a lot of work actually to unpick that and realize what's going on, because you see that, of course, with the NMDA antagonists like ketamine. In low doses it seems to have this quite distinctly, I don't know how you would describe it. I describe it as kind of a dissociative psychedelic at low doses. Some people disagree with me there and say it's not a psychedelic. I would disagree. I think it is. And then you push further and you get this bizarre, very different, highly dissociative anesthetic effects, where the cortical network actually shifts into a completely different type of activity that you wouldn't necessarily have anticipated. So you get these kind of plateaus, and then you can push further and further. And you get a really dissociated and anesthetic, where you get these kind of K-hole effects, which emerge as well. And I think a lot of that is still with many different drugs we don't really understand, and we don't necessarily anticipate the kind of psychedelic effects that you get with certain drugs. You're talking about this one, gaboxidol. I would never necessarily have anticipated that you would start to get psychedelic effects, but clearly you do.
Yes, yeah. Muscimol would be the more common example of a
Right.
GABA A agonist psychedelic.
Exactly. Yeah.
So you discussed a unitive model of psychedelic action, and from a molecular neuropharmacological perspective, there isn't one as far as I can tell, right? You have different types of receptors, different networks of neurotransmitters that are able to elicit this sort of effect. And when people say, well, why are all these pharmacologically and chemically disparate substances capable of eliciting an effect that could be classified as psychedelic? I've never had a good answer to that question.
Mm-hmm.
Do you?
Kind of, yeah. So, well, what I do in the book is to start with, how does the brain construct its model of reality? Or construct your world model? How is that tested against sensory information? So the prevailing model that I work with is a formulation of predictive processing, predictive coding, which is the idea that your brain has this working model of the world. It then uses the model to try and predict the patterns of sensory information that it's receiving. When it gets that prediction right, it basically inhibits that sensory information. So it isn't processed by the cortex, because in a way, your brain already knows about it. It doesn't need any more information if the model is working. But if it fails to predict the sensory information, then you generate an error signal, the difference between the actual sensory information and the predicted sensory information. We get an error signal. That error signal is then passed up through the hierarchical cortical networks and is used to update the model until the error signals decline again. So it's this continuous cycle of making predictions and testing those predictions against sensory information and then updating the model. So that's happening all the time. And that, and your world model emerges out of that process. It emerges as this pattern, this unified pattern of activation of all of your cortical columns, all of those cylindrical columns of neurons that make up your cortex. And that's your world. So that's where everything is unified, if you like. You start from there. Then you think, okay, what are the ways that we can perturb this system? How can we manipulate this system? How can we alter the model? How can we alter the way that sensory information is passing into the cortex? So the flow of these error signals. Can we manipulate the predictions? Can we manipulate the integrity of the model itself? So, and thus its ability to make predictions. All of these things, there are many different ways that you can perturb this world-building machinery to generate what we experience as psychedelic effects. So with the classic psychedelics, for example, you're perturbing, you're stimulating these deep layer neurons, these deep layer pyramidal cells in the cortex, which are important in maintaining the structure of the world, your world model. When you stimulate those, what is basically happening is that it disrupts the integrity of the model. It allows information to start flowing out of these well-defined networks that your brain has sculpted throughout your life and really throughout evolution and your development and your experience. Your brain has been learning to construct this stable model of the world. When you start stimulating these deep pyramidal cells, information starts to flow out of these networks. The model gets shaken up, becomes more fluid, becomes more dynamic. Then what happens is the model starts failing. It starts becoming less able to make successful predictions about sensory information. And so it's less able to quell or to stymie prediction errors, which is the measure of sensory information input. So you get an increased sensitivity to sensory information as well. So that's kind of the basis of the low-level psychedelic effect, with something like a low dose of mushrooms, for example. Then when you start to push it further, you get a stronger perturbation, or with a very particular perturbation, with a molecule like DMT, you start to get a behavior that you see with other complex systems, where instead of just kind of flattening this attractor landscape, so instead of just allowing the cortex to explore new states, which is really what the psychedelic state is, the cortex is moving out of this wells and valleys of this attractor landscape. The landscape kind of flattens out and your brain is able to explore other regions of what I call this world space, this state space of the cortex. But when you take something like DMT, what seems to happen is it's like this attractor landscape collapses into an entirely new geometry. Your cortex, an entirely novel pattern of activity, an entirely novel world model starts to emerge because of this perturbation. Again, we don't really understand why that happens, but it clearly does, because the world that you then experience seems to bear no relationship to the normal waking world. So that's kind of what's happening with DMT. With 5-MeO-DMT, you get a slightly different perturbation at these deep layer pyramidal cells, and this causes a different pattern of emergent behavior to emerge, and this is experienced as these formless white light worlds of the 5-MeO space. So that's kind of the classic psychedelic. But then we can think about, well, what happens when you stimulate with a tropane? So what the tropanes are doing is they're blocking these muscarinic M1 receptors. Now, what are they doing? Well, they are controlling the flow of error signals. So your brain can control these errors. Remember, the error signals is basically when your brain gets predictions wrong about the flow, the evolution of sensory information uses the error signals to update its model, but it can control the volume of these error signals if it doesn't really trust the sensory data. So if you're driving down a road at night and it's really foggy, your brain, the sensory data, it might be quite noisy and messy. So your brain doesn't really trust it. And so it turns the volume down on these error signals. Whereas on a bright sunny day, it trusts the sensory information more. It trusts the error signals. It turns the volume up. And the volume control is partly acetylcholine binding to these M1 receptors, which are excitatory. So they increase the volume of these error signals. So what happens with the tropanes is that they block these M1 receptors, they're M1 antagonists, and so they turn the volume down on all of these error signals. They turn the volume down on the sensory information. It's like you're entering an environment where your brain doesn't trust the sensory information at all. So your world model becomes completely disconnected from sensory information in a way, or from being held account by sensory information, by sensory testing. And so this is why the tropane world becomes unmoored from the external world, and so it can become very, very dreamlike. Phantom smoking, which I'm sure you've seen, where people will smoke cigarettes for long periods of time. You know, what's happening here, I think, in my way of thinking about it, is that there's been some fleeting idea in the head. Did I just light a cigarette? Now, normally you would think, did I just light a cigarette? No, I didn't. But if you get that idea into your head and your brain kind of incorporates this cigarette into your world model, and that's not corrected, you're not receiving the error signals that say, hey, there's no sense information coming between my fingers, there's no curl of smoke. This, all of that would normally correct that model, and you would notice then that the cigarette would be removed from the world model. But here in the tropane state, that's never corrected, and so people will continue to smoke this clearly non-existent cigarette for long periods of time, or people will entertain people in their room for hours on end. They will be kind of having a party with all these people in their room that don't exist. Because the errors aren't created. So they're having a kind of waking dream. So it's a completely different type of psychedelic state, but it's clearly a visionary state. It's clearly an alteration in the world model that is clearly deviated from this normal, stable, adaptive model of the environment. So in that way, it's psychedelic, but the mechanism by which it is achieved is very different. And then get into salvinorin as well. What's going on there? Well, the colostrum is involved. It's this, I see it as like the orchestrator of cortical activity. Has this global inhibitory effect. It's controlling cortical activity. If you stimulate the colostrum, which is connected to almost all areas of the cortex, the person will basically become unconscious, or they will lose their memory briefly. It's like they're not there for a few seconds while you're stimulating the colostrum, because the colostrum has this overall inhibitory effect on the cortex. Now, what salvinorin is doing is binding to these kappa opioid receptors, and it is inhibiting the colostrum. And so you have this like release mechanism. The colostrum is no longer regulating the activity of the cortex, which is allowing the cortex new emergent patterns of activity to emerge across the cortex, which is experienced as the bizarre reality tearing worlds of the salvia space. I describe it as like telling a conductor to instruct the orchestra to play a complex piece faster and faster and faster, and then shooting the conductor in the head. You can just imagine the whole thing kind of goes crazy. That's really, I think, what's happening inside the brain when you stimulate particularly these kappa opioid receptors in the colostrum, is you're applying this release mechanism, you're pulling away this really important inhibitory controller of cortical activity and the control of the movement of the cortex from state to state, just like a conductor of an orchestra. So that's the overall mechanism, the kind of the high level mechanism, I think, of what's going on with salvinorins. So yeah, so there are a number of ways to achieve. Ultimately, it's all unified by you're altering the way that you're perturbing the world building machinery of the brain and causing it to construct a different model, and that to me is that's where the unification takes place. But you're right, there's no unification in terms of the different ways that you can achieve that. There are a number of what I call them switches, really, these multi-level mechanisms that start with stimulating or inhibiting, antagonizing a receptor, but then it has this network effect, subcellular effect on the emergent properties of these subcellular networks, and then ultimately changing the cortical network activity that's actually constructing your world model.
Right.
That's it in a nutshell.
Yeah. I mean, I understand what you're saying, but, and I understand that you can see this as unitive in the sense that these are all disrupting world building, but this fundamental issue of the disparity in mechanisms remains. And why, I mean, I guess that is the answer as far as you're conceptualizing it, that it is different ways of disrupting these models of reality. And it makes me wonder about all these other attempts. I mean, what are your thoughts on Robin Carhart-Harris's work and the default mode network, which is maybe another attempt at somehow, although I don't know how much they've looked at other psychedelics and their patterns of alteration of functional activity in the brain, but is there any neuroimaging unity in these different states? Is there any kind of...
Yes. Yeah, the answer is yes. So the default mode network, this is a very high level network. So this is a network of networks. So the cortical network, as I refer to it, is this highly complex network of these cortical columns. These basically the smallest functional unit, if you like, of the cortex is this column, this cylindrical column of neurons we call a cortical column or a hypercolumn, and these are packed together like a three-dimensional mosaic. And it's the pattern of activation of these cortical columns that generates the information that is your world. However, you also have each of these kind of areas of the cortex, these network of cortical columns also connect to other areas. So you get these networks of networks, and the default mode network is this very high level network that sits at the top of a hierarchy. And the default mode network is important in controlling the activity of other parts of the brain, and has this kind of top-down effect. And with neuroimaging, you can't see, certainly currently, you can't see kind of disruption at the cortical column level. You can't see individual changes in cortical column activity, which is kind of what my model talks about theoretically. But what you can see is the change in the connectivity between these networks of networks, these high-level networks. And so that's why I think Robin's pioneering work really on functional neuroimaging of psychedelics looks at the disruption of these high-level network, but it's the same principle. You've got disruption of the world-building machinery, disruption of these, the activity of these networks, the way that these networks interact with each other, the integrity of the networks themselves. All of these things are disrupted, but fundamentally, it's the change. It's the perturbation at the low level, the level of drug receptor interactions, at the level of connections between neurons. How do the way that neurons share information change under the influence of the psychedelic, which is bottom-up effect, which then affects the way that cortical columns interact, and thus ultimately you start to see global changes in cortical network activity, global changes in the way that these networks of networks maintain themselves and how they interact with each other. So you see disruption. You start to see the default mode network. You start to see reductions in what's called functional connectivity. So when these different parts of the brain are active together, that starts to be disrupted. So you start to see the integrity starting to break down. You start to see networks that were once fought against, that once antagonized each other. They start to lose that ability. So you start to get networks that once didn't really speak to each other. You start to get communication, cross-network communication. But it's all indicative of the same process going on at a much lower level, which is the disruption of this world model. So yeah, that's what you're seeing with a functional neural imaging. It's just a different, because you always have this disconnect between what the pharmacologists are talking about, molecular pharmacologists are talking about, the drug receptor level, and what the neuroimaging people can actually image, and what they're interested in. Of course, they're interested in the high level changes. Why has the world become less stable? Why does it become more fluid, more unpredictable? And you can visualize that using functional neuroimaging. So that's really what Robin Carhart-Harris is doing, I think.
I mean, one thing that I've always had difficulty with a lot of these models of how psychedelics exert their effect is that you can find similar sorts of alterations with non-psychedelic drugs. So people will talk about spinogenesis or synaptogenesis or increases in neuroplasticity. And you see very similar things with drugs like cocaine as well, yet they are not being looked at as drugs. They exert an effect that is totally dissimilar. So do you see that there is something fundamentally different from other drugs in the way that these act?
Yeah, I think so. I think there's limitations in what you're going to observe. You have to you have to understand that there's always going to be a limitation in what you can judge just from looking at patterns of neural activity. And just because you see the same, what appears to be the same kind of high level changes in activity in the brain, doesn't necessarily mean that it's a similar process going on, or the same process going on. You actually kind of drill down deep into the actual changes in cortical changes in neuronal activity or cortical column activity. So they can be quite different, I think, which might explain. I think with advances in neuroimaging, you might be able to tease apart these differences more. But I think that's clearly the case. Clearly, phenomenological studies are also important. People need to be reporting what kind of experience they're having, and how that correlates with changes in cortical activity, and then how that connects to what's going on at the deeper level in terms of measuring activity at a finer grain level. But I think with like salvinorin, for example, there have been some studies that have looked at, quite recent studies, actually in the last couple of years, I think, have looked at the changes in cortical activity under the influence of salvinorin. You can actually see these same kind of effects. So you see disruption of the default mode network. But that's not it as well. You're also looking at changes in neural entropy. So the variation in the patterns of activity, the neural activity seems to become more random under the influence of the psychedelic. So you have lots of lines of evidence at many levels of organization, and the difficulty is to kind of piece all that together and work out, okay, what's actually going on? Can you make that link between a receptor being stimulated, perhaps a single receptor subtype being stimulated in one particular layer of the cortex, and then can you connect that through the levels of organization, working upwards to these global changes in cortical activity? And that's not done so much. That's what I tried to do with the book, is try to walk the reader through what's going on. Everyone knows that the 5-HT2A receptor is important for, it's the crucial, the key locus for the classic psychedelic drug activity. Everyone knows it's an agonist or a partial agonist. And then everyone is also familiar with Robin Carhart Harris's work, where he shows the disruption of the default mode network. But very few people have tried to actually talk you through what's the connection between them. How can we explain why stimulating this small this receptor in this deep layer of the cortex has this effect on global cortical activity. And we're still learning a lot. I have this working model of how I think it works, but no doubt that will need to be updated in the next 5 years or something.
I'm always struck by how, at least in my own work, how little is often explained in the research that I've done. I've spent many, many years studying DIPT, and I've done all sorts of different experiments trying to elucidate the mechanism of DIPT's action. So, injecting mice with DPT and DIPT and looking for different patterns of CFOS expression in the brain. And of course, you would hope that there will be some kind of clear outcome. DIPT impacting auditory perception, well, of course, there'll be increased CFOS expression in the auditory cortex, but there isn't. There's no difference. You could never know. And if you look at the constellation of receptor binding of DIPT relative to something like DPT, not a single receptor stands out that has any explanatory value. I've recently identified a obscure target that actually may explain something. I haven't published it yet, but this is, who knows? It may be that this is also a mistake of some kind. But one thing that I'm struck by over and over again is that with all of the work we have done and with our, with the best tools that we have, I don't think we'd be able to predict a lot of these states based on what we observe in terms of any of the neuroscientific work. We know because people experience these things that this is what they do. But if you didn't know that people experience these things, would you ever be able to predict? There isn't a single scientific paper published anywhere that you could know that DIPT would exert its effect primarily on auditory perception. There's not any evidence for that outside of human use. If it weren't for human use, you would never know that about it. And I think that the same is sort of true about psychedelics in general. Like, do you think that we would know this if it weren't for human experience? Do you think that there's something conclusive in the way that they interact with the brain that would allow you to predict this sort of activity?
I think you could perhaps predict. I mean, using network computational models, you might be able to predict that stimulating a particular receptor, or at least stimulating a certain layer of neurons, might disrupt the model. You might be able to say something very kind of broad and general and vague like that. But I certainly don't think we're at the point where we can say, oh, if you alter this methylation status of this amine, then that's going to make it auditory, or this is going to have this particular type of effect. We're not there. We're very far from being there, because the problem is that we're dealing with a complex system, and I mean that in the technical way, in that your brain and your world model is an emergent system. The function and behavior of your brain emerges from the highest level of a hierarchically organized complex system. Complex systems embedded in complex systems. Your cortex is a complex system that emerges from the interactions between billions of neurons. Each neuron in itself is a complex system that emerges from the multitude of interactions between these subcellular signaling networks. So it's doubly difficult there, or more. It's probably exponentially more difficult then to predict what's going to happen at this highest level of organization when I perturb one of these complex systems at the lower level. It's almost impossible to predict. And then we have all of these different ways you can perturb it. You have all these different receptor subtypes. Each receptor is perturbing the subcellular network in its own particular way. And that might change as well, depending on how you stimulate that receptor. So you have functional selectivity comes into the picture now. It's like, oh no, this just made everything worse, right?
No.
We have this beautiful classical idea that a receptor is you switch it on, you switch it off. It's like, this is perfect. We now have it. And then people come along and say, actually, no. If you stimulate it with this molecule, it switches it on in this way and generates this pattern of emergent behavior inside the neuron. The signaling network changes in this way. But if you use this molecule and the same receptor, it's also an agonist. It's also activating the receptor, but it changes the activity in a different way. So that in itself is like, we can't even predict what's going to happen with the single neuron when you stimulate it with a molecule, because then you've got all these different receptors which might also be tickled to slightly different degrees, and each one has its own functional selectivity profile. So you have a complex system that's being stimulated in a very complex way by one molecule, and then that is from that itself is part of a larger complex system, which is the cortex. So just talking about it kind of makes your head spin a little bit, because you just find these pockets of complexity everywhere you look. And so it doesn't surprise me. It amazes me. It fascinates me. I think it's remarkable. You can change a molecule in such a subtle way and yet and generate completely different effects or highly specific effects, a shift from visual to auditory. That's amazing. But I don't think it's necessarily surprising given what we understand about or don't understand about complex systems, right? And this is where computational modeling comes in. This is where we have to think, okay, we need to start modeling these complex systems. But you're right, we're so far away now. We're still at the stage where we have to. It's brute force. It's finding new molecules and then pumping them into human brains and seeing what happens. We're still there. And I think we're going to be there for some time yet.
And so that brings us to the extended DMT infusion work, which is what you are best known for. And this, I think, is so interesting because it's really out there work. And I'm under the impression that you've started this work, that it's underway. I know you co-authored a paper with Rick Strassman about it in 2016. So could you tell me the history of this project and this idea and where it currently stands?
Yeah, so as everyone is aware who's used DMT, who's interested in DMT, it's the most bizarre and shattering experience. You kind of pop into this space, so you're hurtled into this space, you kind of look around wide-eyed for a few minutes, and then you're kind of dragged back out again. So it's in many ways, for many people, that's more than enough. But there are people that think, wait a minute, this place needs exploring. We need to get to grips with this space. It's not enough to spend 3 or 4 minutes in there at a time. It's like I think of it like a deep sea diver. It's like you need a mechanism to push someone into the space and hold them there, rather than just be limited by 30 minutes on a tank. You want a way of keeping them down at these levels and keeping them alive. So I think of it like that, is what's that transition between normal kind of open water diving and deep sea diving. That to me is the difference between normal kind of standard bolus injection of DMT, 30 milligrams at once, and the extended state. It's thinking about how can we extend the DMT experience for 30 minutes, for an hour, for 5 hours, for a day, if you want, right? So that was the original impetus, the motivation, if you like, of trying to think about this. But then I started looking at the pharmacology. DMT, as you know well, it's remarkable on so many levels, in that its pharmacology is so interesting. And Rick Strassman in the 90s is of course famous for all of the trip reports that became part of DMT, the spirit molecule. But he also did this important study where he injected people with DMT at 30-minute intervals and measured their response on his hallucinogen rating scale to look at the subjective tolerance of DMT. And he found that he was getting the same response every time. There was no subjective tolerance to DMT, even with closely spaced doses. So that, to me, together with the fact that DMT it's very rapidly metabolized, very cleanly metabolized, it just struck me that this molecule, dimethyltryptamine, has all of the requisite pharmacological peculiarities that anesthetic drugs do, drugs for general anesthesia. So in general anesthesia, what you don't do, I mean, I'm not an anesthesiologist, but at a high level, what you don't do is kind of just inject someone with a drug and put them to sleep. What you do is you use an infusion machine. You inject them with a very short acting drug that doesn't have subjective tolerance, and with informed by a pharmacokinetic model, such that you understand how quickly it's entering the brain, how quickly it's being metabolized. All of this is mathematically modeled beforehand, and that gives you an infusion rate that will, in theory, maintain the level of the anesthetic drug in their brain at a fairly steady concentration. So you can keep them under anesthesia in a very controlled way. You can push them deeper. If you're cutting, maybe making an incision, you can push them a little bit deep. You can also bring them out into a more shallow state of anesthesia. So you have a lot of control over this by maintaining and manipulating the brain concentration of anesthetic. So it struck me, wait a minute, DMT has the same properties. It has the same qualities. It should be amenable to using this technology, which is called target-controlled intravenous infusion. That's the standard technology in anesthesiology. So I thought, okay, let me take this idea further. Let's see if we can actually create a kind of a proof of principle model and actually show that this would work in humans, that you could maintain a relatively stable brain DMT concentration. And for that, I know that I needed actual blood data. I needed blood concentration of DMT over time data. And I knew that Rick Strassman had that because I'd seen it in one of his papers that he published in the 90s. So I just sent an email to Rick and said, I'm interested in producing this model and writing this paper. I have this idea for extended state DMT. And, like 30 minutes later, he sent me this old Excel file, which was badly labeled and, badly annotated. But, I was kind of able to make sense of it enough to actually build this mathematical model of the pharmacokinetic model of DMT. And that formed the basis then of this paper, which I published with Rick, which received a lot of attention. It also, both positive and negative, I dare say. Talk about the negative as well if you want, but largely positive, and it spawned a lot of hyperbole. Really, scientists are building a DMT machine to communicate with aliens and this kind of stuff. Just the usual media stuff that went along with that. But that was, in terms of my kind of direct involvement, it kind of, at least, I mean, in Japan, I'm not injecting people with DMT. So please don't email me, people, and say, can I volunteer for this? Because it's not something I am actually doing here. But I knew that the technology should work, and I knew that people would become interested, and they did. So the Imperial College team, particularly, they've taken this the furthest. So they took the idea and really refined the model. They produced a much better pharmacokinetic model than me and Rick were able to produce, one that was actually ready to be deployed in humans. And so this is 2016, I think. I published the paper with Rick. And now they've actually just finished at Imperial College the first study in humans of this, it's come to be known as DMTx. And so they put people under, if you like, or into, held people in the DMT space for about 30 minutes. So it's a small pilot study which should be published next year. The paper should be submitted, I think, towards the end of this year, so this month, I guess. And they're starting to see what I predicted. So I predicted that what you would start to see is that after this initial roller coaster phase, the initial stage is you're kind of thrust into the DMT space, that the brain would start, the state should start to stabilize, because your brain would effectively settle, if you like, into constructing this alternate world model. And that would allow you to then navigate and explore the space, to establish communication with beings within the space, that kind of thing. And there is some evidence that even at 30 minutes, you're starting to see that now, that they go into the space. Yes, it's an initial rush and it's all crazy and wild and chaotic, but then everything starts to stable out. So there's really pleasing evidence, in my opinion, that this can be continued, that you could in fact hold people in there for hours at a time. And then it's like, what do you do with that? There's so much. It opens up, in my opinion, a whole new frontier where you can actually systematically think about experiments. How do we explore the space? How do we explore, do we map the space in some way? Can we think about its geometrical structure? Can we bring information back from the space in real time? There are ways we can do that. We don't have to rely necessarily on trip reports, as Strassman did, and has been the general approach now forever, really, is to talk, what did you see? What did you experience? That to me is very limited. So developing techniques for bringing back information. This is kind of Leary's experiential typewriter back from the 1960s, but bringing that into the modern age. Can we establish communication with these beings within the space? Real time, two-way communication, information coming from the team waiting on the outside, then being delivered to entities, beings on the inside. It's yeah.
And how would this be done? You're saying that this information could be conveyed in a way that is separate from a trip report. What would be that technique?
Yeah, that's a good question. So in the 1960s, so Leary had this experiential typewriter, which was 2 keyboards, one on each hand, with, I think there was 5 or 10 buttons on each keyboard, and it was connected to one of these needle things that drew on the paper. So very primitive, but quite technologically advanced for its time, I guess. But I can imagine something like that, where you would basically be training people to manipulate something in their hands, so they wouldn't have to rely on talking necessarily, although that's not necessarily out of the question. You can train people to speak during the DMT trip, but I think having something more precise, where you can deliver information to the team on the outside, and also to deliver information in the opposite direction. So you might have a mathematician or something who's waiting on the outside, who is asking certain questions about the structure of the space. How do these lines appear? What kind of shapes are you seeing? What is their relationship? And they would then map that to some kind of model of the geometric structure and the dimensionality of the space. I'm not a specialist in that regard, but you can just imagine once you have someone in the space for 30 minutes, an hour, 2 hours, 3 hours, whatever, then the DMT world is your hyperdimensional oyster, so to speak. There's so many different things you could imagine doing. I think we just it's like when we first was able to put someone on the bottom of the ocean. It's like, oh, what can we do now? That's the first part, is getting them there and holding them there. Then you think, wow, so many things we can explore, so many new species we're going to see, all these different things, all these different experiments that we can do now that we have the technology to keep them there. And I see the DMTx technology as that. It's just the technology. It's just the way of keeping someone in the space for extended periods of time.
And I understand that the work hasn't been published yet, but is there anything that you can share about the experiences of the participants in this study? How well tolerated it was, how it differed from a shorter duration DMT experience, how it differed from something like ayahuasca or high dose psilocybin?
Yeah, so dealing with the ayahuasca issue, I think that's kind of important generally, is that people will often say to me, haven't you heard of ayahuasca? You know, kind of, because that's the response that I got in the early days. We dealt with it in paper. I mean, as you know, the breakthrough T experience, if you measure the DMT levels in the blood, it's about 5 or 6 times higher than what you reach with ayahuasca. So these are very different levels. We're not talking about the same thing at all. And of course, the ayahuasca, although it is an extended experience, you're still getting, it's still rising in the bloodstream. It's still undergoing this rise, this plateau, fairly short plateau, and then this long tail. And of course, it's accompanied by vomiting and shitting and all of the other kind of unpleasantries that go along with ayahuasca. So it's not just a chemical ayahuasca trip at all. This is much more precise. You are maintaining a much higher level of DMT in the brain, and it is stable. So you're not relying on the pharmacokinetics of DMT in the bloodstream, coupled with the MAO inhibitor. You're relying on actually keep pumping in real time DMT into the brain to compensate for metabolism and excretion. So it's very different to ayahuasca first. It's not the same at all. And in terms of the experience, there's not much. They fairly tight-lipped in terms of details. Certainly throughout the study they were, because they didn't want to influence other people. But what we do know, first of all, is that the study has been, by all accounts, a great success, and that they can actually use different dose levels, so they're able to actually maintain a stable state at different dose levels. So it's not just breakthrough, it's not just kind of shallow sub-breakthrough levels. They're actually able to bring someone into a breakthrough state and actually hold them there and actually stabilize the state. So I think this first study, what you're going to see is going from theoretical proof of principle, which is what mine and Rick's paper was, to proof of principle in humans. But I think subsequent studies, that's when they're going to start asking, okay, this actually fucking works. We can do this now. We've got a good model. We've got a good protocol in humans, which will be published, of course. So other groups around the world can then pick this up and say, okay, we have a model that works in humans. We can start replicating this. And I think you're going to see groups popping up around the globe that are starting to work on this now. But I think it's going to be subsequent studies where you're really going to see people kind of wrestling with what you actually do now that we have this technology and that it's actually functioning, that we can keep people within the space for hours at a time. What kind of experiments are we going to do? So I think it's tantalizing, I think, at this stage. I don't think we know enough, but in subsequent studies, I think we're going to really start to see what this technology can produce.
It's amazing. I'm excited to see what happens. Are you aware of the historical precedent for this? I mean, reportedly,
yes.
Gordon Todd Skinner would do some version of this with an IV drip in the William Leonard Pickard missile silo lab. Have you spoken with any of the people that underwent his DMT infusions?
I haven't, no. I've read accounts, and I actually wasn't aware of these until after I published the paper, and people started writing to me and saying, "You heard of crystal coal, was it?"
Yeah.
Yeah. And so, I realized then that that people have been, riding the waves of DMT, so to speak, doing this in a kind of a more crude way, if you like. But it's kind of cool to see that this idea has some historical precedent, and people have been thinking about this before. So I don't claim to have this completely novel, amazing idea, but it's just thinking, really what I was trying to do was say, okay, let's bring our best tools to the table, our best pharmacological and clinical tools, and to me that is what you should be doing with DMT. Again, this is where getting into the negatives kind of attention and negative responses to the work. That people were saying, "Oh, this is a sacred molecule. You should be treating it with more respect. You shouldn't be pinning people down and sticking a needle in their arm." They're not pinned down, by the way. But anyway, but to me, I feel the complete opposite. I think if you really take seriously the idea that we could be somehow communicating with some other intelligence, which I don't discount at all, wherever it's from, whatever its origin, whether it's structures deeply embedded in the collective unconscious or some other dimension, whatever. If we really take that seriously, then it's not particularly diplomatic to kind of burst into their realm for 5 minutes, even if you do it facing due east on a hand-woven rug with a lovely hand-blown glass pipe. Lovely. But it's not particularly diplomatic to burst into their space, look around wide-eyed and go, fuck, this is amazing, man, and then bugger off again, as we would say in the UK. That's not particularly respectful, in my opinion. And the most respectful thing to do is to treat it as this incredible technology that it appears to be, and bring our best tools to the table. I think, how can we be much more diplomatic by entering the space, looking around with great humility, understanding we know so very little about the space, and going with an open mind, wanting to learn, wanting to actually establish communication with whatever it is that is in this space. That, to me, is much more respectful a way to deal with DMT than as just some wild roller coaster that you do for kicks, or wild roller coaster because it's amazing. Oh, have you seen this? Take this drug. It's astonishing. That's a good start, dipping your toes into the water like that. But I don't think once we've mastered that, which we kind of have, we've taken that to as far as it will go, vaporizing DMT, we've kind of reached the stage now we can do that quite efficiently. I think really we need to start treating it as this remarkable technology that it clearly seems to be, and approach the DMT space in a much more serious way and sincere way, with a sincere aim to understand, to explore, to spend time in the space, with the beings within the space. Do you agree with that, Hamilton?
Oh, certainly. I mean, I certainly don't think, I would never say that what you were doing is desecrating DMT. I mean, I don't.
Right. I'm sure you wouldn't.
Yeah. That strikes me as a completely ridiculous criticism. What I wonder, and this is not a criticism of the work, but it's just something that I have observed, particularly in the work of John C. Lilly and Marcia Moore, that there can be a sort of seductive desire to understand the space by spending more and more time. If I just take ketamine one more time, then I'll understand ketamine. And if I take it at a variety of doses and take it in a sensory deprivation tank and I keep going back, then eventually I'll understand the space. And this is not to say that you won't gain an understanding, but there's something about it that feels maybe analogous to somebody that becomes very fixated on lucid dreaming in order to understand their dream world. But are they understanding the world of dreams broadly, or are they understanding their own psychology? I mean, there's a suggestion in what you're saying that there is something maybe universal in this space as opposed to personal. And I've always had this idea that was, shared by Alexander Shulgin, that the molecule doesn't contain information. The molecule is not conveying ideas. The experience of the psychedelic is something that is generated entirely in your own brain. And so my question is, do you think that there is something, you talk about the geometry of the space, and I don't doubt that there can be a geometry of the space, but to what extent do you think that this is a universal space versus a personal one?
That's a great question. I don't think we know. I think it's probably a combination. I'm pretty sure that, as I said at the beginning, whatever the nature of the space, whatever the nature of the DMT space, your brain has to construct a model of it, and it has to draw on what the machinery, the patterns that it is learned to construct. And so I think you're always going to see a personal element, a personal aspect, even if the DMT space really is a true other dimension, another reality, you're always going to see personal aspects. Your brain is always going to draw upon the archetypal structures, for example, these deep inherited structures that go way back to the beginning of human civilization. They are, to some extent, going to be drawn upon to try and make sense of the information your brain is going to receive. And this makes it really difficult to tease apart which aspects of the experience are intrinsic to the DMT space, if there is a true other DMT space, some other reality, and which parts are your kind of coloring, if you like, from your own neurology, from your own these deep embedded neural structures. That's the real difficult thing. People assume that if the DMT space is real, then you have to be kind of being it as it is. And of course, no, that's a huge assumption. We don't see the normal waking world necessarily as it is, whatever that means. So it's completely erroneous to assume that we're going to be seeing the DMT space, whatever this space is, as it is. Your brain is always trying to construct a model of it, a coherent model, as best it can. And so clearly there's going to be personal aspects of this. Now, it could be completely personal. That's also a possibility. It could be that you're activating deep embedded structures from your kind of ancient structures, from the collective unconscious, which is really these inherited patterns of neural activity that have been inherited over hundreds of thousands of years. It could be something else, and that's also going to be mixed in with memories and cultural influences as well. So you get this tangled mess trying to work out what you're dealing with here. People say, oh, just because it's culturally influenced, that means it's not real, or that you're not really communicating with some other intelligence, some other space. But that's not necessarily the case, because it's always going to be a mixture, always going to get this heady mixture of both personal and non-personal. And part of the job, if you like, part of the role of this or the motivation of this kind of extended state work is to actually think about, how do we tease those apart? How do we separate the personal from the impersonal, if there is truly some impersonal element aspect to the DMT experience?
And do you see DMT as representing something fundamentally different from other psychedelics? Because, of course, every molecule has its own constellation of affinities and functional activities at different receptors. But I've always seen DMT primarily as a substance that is unique in terms of its pharmacokinetics. I've always felt that many of the states that DMT can produce can also be produced by something like LSD. The difference is that in order to achieve that effect with LSD, you will be incapacitated for an entire day, whereas DMT can allow you to reach that same peak and then return in a relatively short period of time. I mean, that's not to say that there aren't differences between the psychedelics. There absolutely are. And I've tried, DMT and DET and MET and MPT and DIPT and DPT and all of these different tryptamines, and they're all different. But I felt that the major distinguishing factor of DMT, in addition to its profoundly visual effects, is that it's so rapidly metabolized that you can somewhat safely and comfortably go so deep and still return.
Yeah, that's clearly. I mean, people have described. I mean, I've never had what I would call, super heroic doses of psilocybin, but people have described entering these similar kind of spaces, reaching these same kind of states. Kalindi Lee, I think, I can't remember how you say. I mean, he was, like 40 dry grams. Like insane doses of psilocybin mushrooms, and he would describe going to these other worlds in great detail. So I think DMT is not necessarily unique in being able to do that, but as you say, it's what it is unique is in being able to take you really deep, but not having to deal with a 5, 6, 7, 8, 9, 10, 11, 12 hour trip. You know, you can go in and out very, very quickly and very, very cleanly. That's what makes DMT, I think, unique. And that's, of course, what makes it amenable to this kind of extended state protocol, is that you can bring someone in there and you can maintain the level, or you can bring them out, you can push them deeper. If they say, I want to go deeper, this isn't deep enough, you can push them a bit deeper. If they say this is too much, you can bring them out quite quickly in quite short order. Within, you know, really a few seconds, you can start to bring someone out of the state and have much more control. You just can't do that with the other classic psychedelic. You certainly can't do it with LSD, and you can't do it with psilocybin either. But with DMT, you have this unique opportunity to be able to push someone really as deep as anyone would want to go with the psychedelic, and yet maintain this control over the depth and over the duration. It's kind of a remarkable thing that we have, remarkable tool, I think.
I agree. Yeah. Yeah. And yeah, I mean, I really recommend people listening to this check out your book. I also just appreciate that you chose to make it accessible to a broad audience, that it isn't presenting itself as an academic text, because I think that there's a lot of insecurity in the world of psychedelics where people desperately want to show that they're serious. They're not one of those hippie types. They're not just trying to get high. They're serious academics. And so in an effort to look as serious as possible, they sometimes make their work dry and inaccessible. And I appreciate that there's an artistic dimension to what you did, that it's designed to be beautiful and accessible to people. And I think it's a really valuable thing to do. So thank you. Thank you.
That's very kind words. Yeah, that's what I attempted to do. I also have a course on YouTube, psychedelic neuroscience master course, which I also go through. It's like 8 units. I go through the neuroscience, taking people through. I don't assume anything, because there's so many people interested in psychedelics who don't have degrees in pharmacology or in chemistry or in neuroscience, and you kind of need a bit of all of those to really understand psychedelics. So I knew there was a gap in the market here. There was this space that just was begging to be filled. I read so many bad books or bad explanations of what psychedelics are doing, even from academics. And so I thought, no, I need to write a book that will actually allow anyone with an interest. It's not an easy read by any stretch of the imagination, but it's perfectly accessible to somebody who really wants to understand and willing to spend the time going through, from all the different levels. So I talk about chemistry, as you say, what's interesting about the structure of these molecules, their pharmacophores, how do they interact with receptors? How does that change the way receptors function and behave? How does this change the way neurons function and behave and interact with other neurons? And how does that change the way that the world model is constructed? And why does that have the psychedelic effect? So, I start the low level and build upwards. So I think by the end of the book, you have this really both broad and deep understanding of psychedelic neuroscience. I don't think there's any other book available that will give you that kind of level of understanding.
Yeah, I don't think so either. And for anyone listening to this that wants to learn more about your work, where should they look? What's a good place to follow you and find your research?
Yeah, so my website is alieninsect.net. There you can download the first chapter of my book, Reality Switch Technologies, Psychedelics as Tools for the Discovery and Exploration of New Worlds. They have the video, but anyway. And you can find links to a lot of my interviews and my paper you can download there. So that's kind of my main base. I post regularly on Twitter as well. Alien Insect, again, is my Twitter handle. I have a Substack, Alien Insect on Drugs, which I post every week at the moment. Again, writing about the science of psychedelics. If you want to buy my book, then you can do that directly from my website, alieninsect.net, or you can just go to Amazon and Google search for Reality Switch Technologies, or you can also look at my highly speculative metaphysics book, first one, Alien Information Theory. I think that covers all the bases.
Yeah, thank you so much. This was great.
Thank you, Hamilton.